All right, so thank you, first of all, so much for joining me here today to listen to me give my thesis defense, really, on my project, Managing Eurasian watermilfoil with Burlap Barriers, and then the response of biological communities and nutrients. So thank you to Kyla for my lovely introduction, and let's get started. So first of all, I'm going to give you a little bit of a lay of the land of how I've set up this presentation, just so we're all on the same page. So I'm going to start out with some background on why this is important and why this project is important, and knowing, you know, um, the response that these communities and nutrients have to burlap. And then we're going to kind of jump into the data, and I've broken this up into two different sections. So I'm going to start with my water quality data, and that'll be part one, and then we'll jump into my community data, which will be part two. I'll end with some conclusions, and then of course I'll open up the floor to any questions that you have. So with that, let us dive in, pun intended. So, as you all know, lakes provide many different ecosystem services. And if I were to ask you to think about, why lakes are important to you, if not all, most of them would be considered ecosystem services. Now, generally, these are broken up into four different categories, and I'm going to run through those really quickly. So first of all, provisioning would be things like food and drinking water, subsistence. Then we have regulating, so that would be things like flood mitigation. We have cultural services, so that would be recreational. So anything like fishing, boating, swimming. And for many groups, lakes are a big part of their spirituality and their culture. And then finally, we have supporting services. So those are going to be those services that help to keep these other ones up and running so that we can enjoy them, right? And that would be things like nutrient cycling within the lake and biodiversity. And speaking of biodiversity, that's what we're going to focus on today, and why, my project is really important. So as we know, to keep all of these things running and so that we can all continue to enjoy these ecosystem services, having a really healthy lake is important, and biodiversity is really important to that. So what the biggest threat right now, though, to biodiversity are invasive species. And by definition, an invasive species is a non-native species that has been introduced outside of its range, and it has the potential to cause harm to the economy, to the environment, and also to human health. So for lakes, aquatic invasive species such as Eurasian watermilfoil really threaten the ecosystem services and threaten that biodiversity, which is why controlling EWM, or Eurasian watermilfoil, is such a major concern. So a little bit about EWM. So it was introduced, it's recorded that as early as the 1800s, EWM was introduced into the Chesapeake Bay area through ballast water from ships. But the earliest confirmed identification was in the 1940s, and since then has spread to almost all of the states within the continental United States, but except for Wyoming, which is interesting. But anywho, so it is really taken hold, especially as we can see of the Midwest right there. But why is it such a great invader, and what are some of the reasons that we don't want it, right? So number one, it is such a great invader because it can rapidly reproduce by fragmentation. So any kind of wave action can break off the tops of the plants or different parts of the plants, and then it can then spread throughout the lake and establish new patches, new areas. And so again, it can really take over a lake pretty quickly. It can also form these really dense canopies at the top of the water that will shade out our lower, more native plants, and so they can't get light and they will die, really creating, again, lowering that biodiversity that we so want to keep for the lake. And then with that, it has been reported in some studies that underneath those canopies you can see lower DO, or dissolved oxygen, which in turn can increase nutrient release from the sediment. And of course, as we know, too much of anything is not a good thing. And sometimes that increase in nutrients, and many times, can lead to, things like algal blooms, and specifically harmful algal blooms, which is something we don't want, right? So again, controlling it is very important. And the current methods right now that are most popular are using chemicals like herbicides and then harvesting with like mechanically. There we go. So think of like a giant lawnmower on the lake going in and cutting, these plants. Now, some pros to this are they can cover a pretty large area with at lower cost. So chemical, but whew, sorry guys, I'm getting ahead of myself here. All right, so anyways, they can cover a large area and are relatively low in cost. Now, some of the more recent, issues that we've seen with chemicals and some of the cons are that with extended use of chemicals, there are some research that has seen, increased hybridization with our native plants. So with our native milfoil, so northern milfoil. And with that hybridization, they're becoming more resistant to the chemicals that are designed to target specifically EWM. Now, harvesting, again, as we talked about, fragmentation is the preferred method of reproduction for Eurasian water milfoil. And so with harvesting, you are going to get a lot of fragments. And while some of these boats will pull in what they cut, or a good majority of what they cut, there's still quite a bit of fragments that are floating about and can spread and, again, take hold. So alternatively to that, there are burlap benthic barriers. Now, benthic barriers in general aren't new to the scene. They've been around roughly the 60s, but they were synthetic. So they're synthetic. So they're made out of like a tarp-like material. They're deployed into the lake to lay right on top of the sediment over the plants, shade out the light, and get rid of whatever's underneath. So again, but then we have burlap barriers, so they work in a very similar way, but instead of using synthetic materials, it is natural, it's hemp, it's burlap, and it will naturally degrade. And then also, again, another advantage is you're not having to use chemicals. So the purpose of my study, with this in mind, was to answer the question, do burlap barriers alter environmental conditions during its deployment? So my study site to do this was at the lovely Lake Leelanau on the Lake Leelanau Peninsula. I'm sure many of you are familiar with this lake. It is made up of two different basins. So we have the northern and the southern basin, and it's hydrologically connected by a narrow channel, aptly named the Narrows. Now, my specific project was in South Lake Leelanau because to date, there have been no sightings of Eurasian water milfoil in the northern basin, which is fantastic. And so yeah, so southern lake it was, southern basin it was. So yeah, so if you'll notice, I picked 3 different treatment sites in 2022 when the Grand Traverse Band of Ottawa and Chippewa Indians and the Lake Leelanau Lake Association reached out to GVSU to collaborate with us on this project. So in 2022, I selected 3 different treatment sites, and that is marked using my yellow bubbles with the stars here. And then I had within that, there are 2 larger areas that were going to be covered, and then 1 smaller area. Now, you'll notice on the north and south ends, respectively, are blue bubbles, and those are marking the 2 reference sites that I selected. And these are sites that are completely native, contain native vegetation. So just something to compare these treatment sites with throughout the extent of my study. So a little bit of how I picked my sites. So we had a little bit of change throughout the year, just to due to logistics, time, people, But anyway, so initially we had an aerial photo of the lake and the treatment areas, and it was gridded, and we randomly selected 3 sites, regardless of size of barrier on each treatment location. And so when we went out there, we would toss weighted markers, and that would where our coordinates told us to go, right? And then we would sample them. Now you'll notice to the left, there's a little patch of vegetation that I made over to the side. And this area, so these spots were used as our reference sites for the water samples only in 2022. But I'll elaborate on that a little bit later. So things changed up just a little bit as far as like selecting sites in 2022 post barrier. After those barriers were laid, we were unable to get back to those coordinates. So instead, we moved along the top of the barrier by boat and then randomly tossed weighted floats over the sides, and those were our sample locations for that sampling event. And again, the reference sites only for water were barrier adjacent, and they were still within native vegetation. So yeah, so that was 2022 post barrier, and again, 3 sample sites regardless of size of barrier. Now, starting in 2023, I did increase the number of sample sites, and our larger barriers instead of 3 had 10, and our smaller barriers 4 instead of 3. And that was just to get a more representative data set from each barrier, based on a size. So we continued to sample those sites and went back to those same sites all the way through to 2024. So for the rest of my project. So reference sites, again, 2022, we only sampled once that summer from reference sites, but we would, instead of very similar method from the boat, randomly tossing weights, but we anchored kind of in the middle and tossed randomly around the sides. Starting in 2023, same thing to select the sites, but again, we did use those same sample sites throughout the remainder of the project. And the reason we were able to is because we had some advanced GPS that we had brought in, and so we were able to more accurately mark those sites and then get back to them. So now let us get into part 1. So we're going to be talking about water nutrients and some water quality as well. So my 2 main questions concerning water quality was, number 1, do burlap barriers affect nutrient concentrations at treatment sites? Was there a difference above, below, after the barriers were deployed? Number 2, are there differences in nutrient concentrations at treatment sites compared to those at reference sites? So what was that looking like? Were the treatment sites so different? So my objectives to answer those questions, number 1, so I wanted to quantify any changes in nutrient concentration after barrier deployment. And then number 2, I wanted to determine if there were any differences between reference and treatment sites before and after the barrier was deployed. So I hypothesized that for nutrients at the barrier, at the treatment sites, that there would be no significant change in nutrient concentrations below the barrier, and that would be due to the gas exchange through the barrier. And then number 2, that there would be significant differences in nutrient concentrations between reference and treatment sites initially after barriers were deployed. So a little bit of info on how I sampled. So pre-barrier and reference sites, so this is before the barriers were laid. So above the sediment, I took above the sediment samples. So all samples were taken using scuba. So we would dive down and get within the plant bed and then take our water samples from right above the sediment. And again, this was for both pre-barrier and reference sites. For treatment sites, post-barrier, so after that barrier was laid, we wanted to look at nutrient concentrations below the barrier as well as above and see what was going on there. So we used 2 large bore syringes to get in between the weave of the barrier. And then for above the barrier, again, very similar to our pre-barrier sampling and reference site sampling, just right above that barrier, we took our water samples. In addition to water samples, I also took sediment samples. So I took 3 ponar samples. So that is just this giant claw that goes into the sediment and gets us a sample. Ponar samples, pre-barrier 2022, and then post-barrier 2024 at both treatment sites and reference sites. And this was just to look at the total phosphorus. We wanted to see if there was a difference pre-barrier versus post, right? And then also I used DO loggers, so dissolved oxygen loggers, to kind of see what happened after a barrier was laid. So we were able to place one of these loggers underneath a newer barrier in 2024 and see what happened underneath. Was it becoming anoxic or void of oxygen, or was it able, or did we see movement? Did it remain well oxygenated? And then we also were able to put one under an older barrier that was laid. So my original barrier laid in 2022 at Grants Point. And so this barrier had been there since 2022 and it was pretty well intact. And so we gently lifted up the barrier and slid it under there so that it could monitor. Now these were both left for 2 weeks during the same time period. So we selected these nutrients. So I selected these 3 nutrients to monitor ammonia, nitrate, and soluble reactive phosphorus, or SRP, because I anticipated changes in oxygen below the barrier. And these 3 nutrients could kind of tell us that story, could tell me that story. So again, we would see if that were to happen, an increase in ammonia, a decrease in nitrate, and an increase in SRP. So results. What did we get? So for SRP, it was soluble reactive phosphorus. It was generally below detection, both above and below the barriers at reference sites. So what that means is that, the test that we used, it didn't necessarily not detect anything, but it could not accurately give us a number because it was so low. So because of this, I was unable to do stats or to perform stats on this data set, but it does tell us a lot, and we do gather a lot from this, because even though we didn't get anything, we should have seen, or we expected to see kind of a spike in SRP if it was becoming anoxic or, again, devoid of oxygen underneath, and we did not see that spike. So that kind of suggests that it's still pretty well oxygenated underneath the barriers. So there's a lot here going on. So these are my box plots, and what I did was I broke them up by site, and I did this because pretty early on, it was apparent that, the sites kind of did their own thing to an extent, and I really wanted to make sure that that was seen. So you'll see I have Maybrick Creek, Grants Point, and Gordon's Point. Now, ammonia, as you can see, big picture from this, we're looking at within each year, or within each sampling event, well, year, and we're looking at above barrier versus below barrier versus reference site. And so what I'm comparing are these middle lines. So I'm looking at the median concentration, not the average, just because the way the stats ended up, the data ended up. But what you can see, big picture, is that ammonia was generally, it varied depending on site, specifically 2023 and 2024. So that's what I want us to gather from these. Okay. Now, I also wanted to look at across years, because I was like, okay, well, ammonia increased under the, here, we'll go back for a second. So ammonia increased under the barrier and was generally higher than above barrier. So I wanted to look and see, okay, well, maybe this is a bit high under the barrier, compared to above or below barrier than it is above. But what does it look like across years? Is it staying consistent, or is it increasing or decreasing, right? So what we did was, so I did this, and ammonia, and what I got was ammonia tended to increase post-barrier 2022, then decrease was the general theme, right? Except for MC. But as you'll see in just a moment when we get to nitrate, Maybrick Creek was kind of a different, a dark horse, if you will, in a way. Lovely site, but a little different. So looking at below barrier, again, here we go, big picture. Again, I'm comparing below barrier across years. So we're looking at the white boxes across the year by site. And ammonia was generally highest post-barrier 2022, but then decreased. And again, except for Maybrick Creek, which again is its own thing. its own. So moving on to nitrate. So this is, again, we're looking at above versus below versus reference, and we're doing this by site. So nitrate was relatively similar among sample sites. And again, except for at MC, which was quite a bit higher than our other sites, just in general. But again, we wanted to see across years what we have. So now looking at above barrier, it generally decreased at treatment sites post-barrier 2022 and then came back up, increased starting in 2023, except for at Maper Creek, and everything kind of remained similar as far as concentrations go. Now, looking at below barrier, nitrate generally decreased, and this is across years, nitrate generally decreased at treatment sites post-barrier 2022 and then increased. But again, if we say it in unison, except at Maber Creek. So what we did learn, and I'll kind of touch on this in a moment, like towards the end with our conclusions, but Maber Creek just in general had higher nitrate than at my other sites. And what I did find out was, this site is very near to where Maber Creek inputs, right? That's why we named the site Maber Creek. But there's quite a bit of nitrogen coming into the system right there. And so this could help explain why we're seeing these much higher nitrate levels at Maber Creek than at Grant and Gordon's Point. But in general, kind of remain the same throughout our study, or remain consistent, if you will, within that site. So sediment results. So we looked at reference versus treatment, and those were significantly different as far as total phosphorus averages. But when you look at treatment site pre-barrier versus treatment site post-barrier, those were not significantly different as far as concentrations go. And the same with reference sites, which suggests that the barriers did not cause an increase in total phosphorus, which again, we're kind of talking about the release of more nutrients and plant death. So we didn't see a significant change in those concentrations. Now, these are two of my favorite figures. So this is from my DO loggers, so dissolved oxygen, and you'll see they kind of tell a very different story. So the one to your left is going to be from under the barrier at 2022, that was laid in 2022 at Grants Point. And you'll notice that we see this, nice spike in dissolved oxygen, and it's got this nice pattern going up and down, right? And then around July 30th, it falls back down to nothing or just a little bit of oxygen, right? Okay, so we see that. Now, if we look at 2024, we see this lovely pattern of, oxygen coming up in the daytime, oxygen levels rising in the daytime and going down at night, which is pretty typical, pretty standard as far as like that pattern. Now, what this tells us is, the barrier at Grants Point, it had been there since 2022, so we're looking at 2 years, right? And there was a lot of sediment that had accumulated on the top of that barrier. And so when we did put the DO logger underneath, we shook some of that sediment off, and so the weave was then, exposed. And so that's my theory, is that that's why we see this, our oxygen levels as they are, because it was oxygenated. But because the sediment, there's a lot of sediment down there, it all settled right back down, then that's when we see it kind of covers up that weave, and we don't see it's not very well oxygenated underneath. While this newer barrier, again, didn't have any sedimentation, and oxygen flowed pretty freely, it would seem. We did get some really low numbers, but it did go back up in the daytime. So let's put all this together and answer our questions. So the question, do burlap barriers affect nutrient concentrations at treatment sites? Well, what we got. Effects were generally site-specific, and then when there were changes, they were primarily underneath the barrier. But throughout time, we did see that concentrations tended to return to pre-barrier conditions. Now, to answer our question number 2, were there differences in nutrient concentrations at treatment sites compared to those at reference sites? Well, initially, nutrient concentrations were not different, actually, immediately after the barrier was laid. And then above barrier ammonia concentrations differed from reference sites, but nitrate did not, again, except for MC Mayberg Creek, as we know. Looking at below barrier, ammonia concentrations, they did generally differ from reference sites, but again, nitrate did not. So really, ammonia was our main nutrient that fluctuated. We'll come back to a big conclusion at the end with everything tied together. So part 2, let's move into community. So my questions regarding communities. So number 1, are burlap barriers an effective management method to control EWM? And number 2, do burlap barriers alter macroinvertebrate and fish communities while the burlap barriers are in place? So my 3 objectives to help answer these questions. So number 1, I was going to evaluate, or I did evaluate the effectiveness of barriers as EWM control, and I observed whether or not macrophytes recolonized the area. Number 2, I wanted to quantify any short-term changes in macroinvertebrate communities after barrier deployment. And number 3, I wanted to evaluate fish community response to barrier placement. And so again, my hypotheses, what I thought would happen is number 1, EWM growth through the barrier would be insignificant because EWM relies on fragmentation to establish new populations, as we talked about. And then number 2, barriers would cause a decrease in the density and diversity of macroinvertebrates due to changes in substrate. And then number 3, there would be a shift in fish community composition due to the barrier placement and what I was thinking was an edge effect. But we'll get into that a little bit later. So just to kind of go over how I sampled. So for treatment sites in 2022 pre-barrier, so I used a quadrat. So I used quadrats that were 0.5 by 0.5 meters, and I collected all plants within that quadrat to identify and count. I did stem counts. For macrophytes, for macroinvertebrates, I used that same macrophyte sample that I had collected for plant identification, and I pulled all the macroinvertebrates off of it to count and identify. And then same with the sediment sample that was also used for total phosphorus. I took the macroinvertebrates out of that as well, counted and identified. For the fish surveys, I used 2 different types. So I used a FICENet survey, and then I did a diver survey. So the FICENets, if you'll turn your attention to the right of your screen, where I've made this lovely plot of greenery. So those 2 slender rectangles on either side are representing the FICENets, and that's where those were placed at the north and south end, respectively, of the patch. And those were left for 24 hours. And then when we went to collect them at the end of that stint, fish were counted and identified. Now for diver surveys, that was done obviously by a diver, and we placed a transect in the middle of the treatment area, and then as I swam down the treatment area, that transect, I counted and identified fish that I saw on the barrier, just to get a good idea of what was going on kind of everywhere. Now post-barrier 2022, instead of a plant collection, I used video within that quadrat. And that was because if anything had grown or started to grow through the barrier, I didn't want to disturb that. And then I did FICENet survey of fish surveys again post-barrier, and again FICENet diver survey remained the same method. So post-barrier, it's looking very similar, but we did have in 2023, we again did the quadrat videos, and then I did a transect video. So along that same transect that I used for fish, I took a video just to kind of get some visuals, right? And get a better idea of what was going on over the entire area. And then for macroinvertebrate samples, if there was vegetation, again, we didn't want to disturb that. So we took a different form of substrate that kind of replaced, to an extent, the vegetation that was there earlier. And so I took the burlap and then pulled the macroinvertebrates off of that to count and identify. For fish, again, same survey types, FICNet, diver survey. And as you'll see from my elongated black boxes, that is kind of where those FICNets were placed, right at the edge. In 2024, everything remained the same. The only difference is with macroinvertebrate samples, I took a sediment sample as well, which again, we kind of discussed earlier, which was used for total phosphorus testing as well. And then there was one other difference with our diver survey. I did include, for larger barriers, I included the offshore and onshore transect, an offshore and onshore transect, just to, again, get a better picture of what was there in that large area. So reference site collections, very similar, especially to pre-barrier. So I did quadrat collection, macrophyte, so plant collections every year, and they were, counted and identified. I did try to do video quadrats, same as the barrier, but it's a little too much right there to like count everything accurately. So again, I took physical samples to do that. For macroinvertebrates, again, same kind of thing. We took them off of the macrophytes that we collected, and then also the sediment pre-barrier, and then in 2024. And then fish remained the same throughout. FICNet survey, diver survey, all remained the same. And we use the same method as with our treatment sites. So let us jump into the results. So looking at macrophyte relative abundance, there's a lot going on right here, a lot of fun, pretty colors. But if you'll turn your attention, so 2022 pre-barrier, as we can see, EWM was that dominant species, it's that orange color, species at pre-barrier sites, or pre-barrier treatment sites, which makes sense. That's why they were being covered with barriers. And then we'll look and see that if you look at KP and PHP, those are our two reference sites. We have a variety of different types of vegetation, which is exactly what we expected. Now, post-barrier, of course, there were no plants and snow bars, and we did not do another survey of our reference sites that year. Now, in 2023, we did, so in 2023, you can see that reference sites, those first two bars, remain the same pretty much, lots of different types of plants. And then our treatment sites, we're getting more native plants. We still do have a bit of EWM in there, but again, just from what I saw and what I observed, those were from fragments and not from underneath the barrier. And then MC, we didn't have any there. So again, we see in 2024, none of my sample sites had EWM present in the quadrat, and you'll notice GP, so Gordon's Point doesn't have anything, but it had some barrier maintenance that was going on, so we didn't get any in our specific sample sites. So big take home here, again, after barrier deployment, EWM continued to decrease, and then our two main plants that were usually always there was our Valsinaria americana and our Potamogeton amplifolius, which is our pondweed, and our eelgrass. And they were usually the first to reestablish. That's what we can see from this lovely rainbow of a figure. So macroinvertebrate densities. So the main thing I want us to gather, or take from this graph. So treatment and reference sites was significantly differed, but only in 2023 when zebra mussels were included. Now, as we know, and as we'll see, zebra mussels really love vegetation. They don't love the barrier. So that is why we have this and there's tons of them. Once you find one, you'll find a million. And they love the vegetation, especially the native milfoils and just those really structurally, lost my word. Anyways, they love vegetation. That's it. But anyways, but as you can see, they if we take those out, they are say it's actually the treatment sites have more macroinvertebrates. So when zebra mussels were not included, as I kind of mentioned earlier, were not included, there was no significant differences between treatment and reference sites. So all remain the same, even though we had that different substrate. Now, looking at macroinvertebrate relative abundance. So big picture, take-home points, zebra mussels dominated vegetation. Again, as I mentioned, if you look at EWM and native vegetation, that dark green is going to be will denote our zebra mussels. And then zebra mussels and chironomids, or little midge larvae, dominated the sediment. So those two, the R sediment and the T sediment, so it stands for reference and treatment sediment, you can see that chironomids really did, both zebra mussels and chironomids really did kind of share in the dominance. And then if you'll turn over to burlap, it was dominated by chironomids in 2023 just because of the structure that it had and where it was located, I believe. But it became, in 2024, you can see it becomes more similar to sediment, which kind of going back to the dissolved oxygen figure that I chatted about, we're looking at that one that was laid in 2022. It just had kind of become another layer of the sediment, right? Because it falls back down on it. So again, this is another figure that kind of illustrates the burlap just reverting back to and going back to that sediment. So macroinvertebrate diversity. So what we see with this graph, so vegetation in general had a lower diversity score than sediment and burlap, and that was because zebra mussels just really dominated that community. And then we see the burlap diversity scores became more similar, again, as I mentioned with the last slide, more similar to sediment by the end of the study. So when I looked at this and I did my stats on this, there were significant differences in diversity between EWM and the sediment. So, which again, they are very different. And that first year, EWM really did have a lot of zebra mussels present. And the native had some, but not as many. But we can, if you have a question, I've got my theories. We must move on. So macroinvertebrate community composition. This is another one of my favorite figures. But I ran an Adonis pairwise. So this is just comparing different communities and associated with, so the different substrates and which macroinvertebrates were associated more strongly with those substrates. So really what we see is that this pairwise, this comparison determined that only native vegetation and sediment types were dissimilar. So you'll see over on the right, this little, this nice cluster of our burlap and our sediments. And then if you'll look to the left, those two green colors, so our more Kelly green and our more forest swamp green are pretty grouped pretty strongly together. And again, chironomids are strongly associated with the burlap and the sediment, and zebra mussels more so with the vegetation, which we kind of have covered. Now, zooming in a bit, this is just looking at vegetation. And then breaking it up a little bit. So EWM is going to be that pinkish color and native vegetation is going to be the bluish color. Now, when I ran this pairwise, so this comparison, it determined that similarities were only between EWM and native vegetation 2022 and native vegetation 2023 and 2024. So while they're similar, there was some differences. Specifically in 2022, we had some differences. So macroinvertebrate community composition on sediment and burlap. So again, a little bit of a closer look at that from that first figure that I showed you that was similar to this, but had everything on it. So this is just looking at the sediment and burlap cluster. So when I ran that test to compare, community composition, it determined that the burlap 2023 was significantly different from all other substrates and years. Treatment sediment 2020 and treatment sediment 2022 and burlap 2024 were significantly different, but that was most likely due to zebra mussels and chironomids, right? Because we're looking at the composition and the different, like, amount of zebra mussels versus amount of chironomids. Sorry, my dog. So moving on to fish. So if we're looking at fish relative abundance, so rock bass were the dominant fish species, as we can see. So they're that green. They really ran the gamut. There were minor changes in relative abundance pre-barrier to post-barrier 2022, and there were minor changes in pre-barrier to post-barrier 2023 and 2024. Now, spring surveys were relatively similar, as we can see, and then in summer surveys, there was minor shifts, and we did see a decrease in yellow perch and yellow bullhead. And also walleye and yellow perch decreased from 2022 summer surveys to 2023 and 2024. So those were our main differences. But again, as we see, little variation. Again, rock bass really took over there. So if we look at this at a community composition, so my comparison determined that only that EWM versus the treatment sites and reference sites was significantly different. And that's probably because we did see in that, you can notice down here, so the EWM is going to be our 3 green dots down there, that they were kind of clustered, but a little bit farther away from the pack. So there were some differences, right? And then looking at our diver surveys, spring and summer surveys, so this is fish abundance again, relative abundance. So spring and summer surveys differed greatly, as we can see. And then from pre-barrier to spring 2024, community became more similar. So again, and these are dealing with, you'll see that it looks like it varies a lot, but this is based on, I didn't have as many numbers, or yeah, there were lower numbers of fish on the burlap, but this is kind of what we got. So let's bring it home. So number 1, let's answer our questions. Are burlap barriers an effective management method to control EWM? Well, yes. It is an effective control method, and it does allow for regrowth of native macrophytes from our study. Number 2, do burlap barriers alter macroinvertebrate community? Community composition was altered with a change in substrate, and then as burlap aged, community composition became more similar to sediment. And number 3, do burlap barriers alter fish communities while the burlap barriers are in place? After the burlap barriers were deployed, fish communities became more similar to that of native vegetation, but again, there was very little variation. So, big take-homes. Nutrients. So variation in nutrient concentrations above and below the barriers were site-specific, as we saw, which is why I broke everything up to compare and kind of look at and show you today. But however, nutrient concentrations were relatively low at all sites. Now, macrophytes with regular maintenance, burlap barriers seem to be an effective control method for EWM while also fostering native macrophyte growth. And then macroinvertebrates. Total abundance of macroinvertebrates at treatment and reference sites were generally similar, and then changes in species composition at treatment sites was primarily due to our zebra mussel and chironomid populations. And then looking at fish, big wrap up, fish communities at treatment sites were similar to reference sites in both 2023 and 2024. So I know it's a lot of information, but I will open up for questions. A huge thank you to the Grand Traverse Band, the Lake Leelanau Lake Association, and all of these lovely people that I've mentioned below, and countless more. But yeah, so I will take questions now. I know that was a lot. Thanks for hanging in there with me.
Thank you so much, Anna. Yeah, if anyone has any questions, please feel free to unmute yourself at this time and go ahead.
Hi, my name's Ron Ray, and I'm a member of Crystal Lake Watershed Association. A couple of questions on the burlap. Does burlap sink?
Um, it does, but it takes a minute. So to lay the burlap, I guess I forgot to say. So there are giant rebar, like there's rebar that is attached to the burlap to help it sink, and then there's also divers that help pull it down. And then burlap sandbags are laid on top of it. But yes, it does sink, but if without help, it takes a little bit longer.
Okay. And then the other question is, any indication of the effect on quagga mussels?
Um, Not with my study, no. I mainly saw and identified, and by mainly, I only saw zebra mussels with my study. I'm not for sure. I don't know.
Thank you.
There's a couple questions in the chat. One of them is, how long do you anticipate it takes for the burlap to decompose?
Yeah, so some study, there was one study that had said, a year or two. But with my study, I found 2 to 3 years, and it really depended on where it was in the lake and kind of what the exposure looked like. So specifically Maybert Creek, it was a little shallower, and it was, you had more light exposure, or a little bit more wave action, woody debris everywhere. And so it pretty much started to really disintegrate and really decompose after about a year. Um, so yeah, but then I had these some deeper sites that took, it's still at in 2024 that were pretty well intact, but starting to go, right? So I can't, it really just depends on where it is in the lake, but I would say roughly 2 to 3 years.
There also a question, and I think you kind of addressed this as, in your slides, but just to clarify, did native plants grow back on top of the burlap, or was it because the burlap was removed?
So the burlap was not removed, so it stayed in. But yeah, so we did see there were some. I thought that it was in the slideshow, but I guess I took them out. There are pictures. So it's coming. So native plants did come back, start to come back, and they would come up through the barrier, through the weave of the barrier. So yeah, so they did come back.
Someone also asked, and I'm sure you might not be able to answer this since you were just doing the research and not the actual deployment, but someone asked, how does cost of using burlap compare to the use of chemicals?
Yeah, that would be a lovely question for our friends at the tribe and the Lake Association. I'm not super for sure on the money side.
Any other questions? Oh yeah. Uh, Someone wanted to clarify who actually did lay the barriers. Was it the association,
It was the tribe,
the tribe?
the Grand Traverse Band. Yes.
And Dr. Lattimore is asking to clarify that native plants came back, but EWM did not. And she also said, why do you think you saw what you saw with the plants?
Yeah, so as you saw from my composition that I made up, so initially after the barrier was laid, we saw no plants, right? And then that next year we did have a bit of EWM come back, but from my observations, it looked more to be from fragments. And that's just because you can kind of tell they're growing top down rather than up through the barrier. Um, but yes, native plants were the only ones that I observed coming up through the barrier. Yeah, hope that answered that.
Yeah. Um, Going forward, how often do you expect this may need to be done to a lake compared to repetitive chemical treatments? Long-term health would need strong prevention efforts alongside, probably.
Yeah, it's very now, my study is focusing specifically on Lake Leelanau and what I saw there, but based on what I've seen there, like, it very much depends, as we kind of saw, it's very site specific as to how many, maintenance and what goes into that. But honestly, as I've been working with these groups, it's less and less every year. So once you get a hold of it, it seems like it's working really well. Um, but yeah, so that's based on my observations and my study, what I've seen.
And can you remind us what the water depth was for the study sites?
Yeah, so it was about, actually, let me think. It's in my thesis. So it was roughly 6-ish meters for two of my sites, and then one was pretty shallow, coming in at about 2 meters roughly. So one was quite a bit more shallow than the other two. And then those reference sites are also going to be, they were deeper as well, so like 5-6 meters.
And a question, if the burlap is not removed, is the rebar removed?
The rebar is removed. I should have mentioned that. Yes, the rebar is removed that day, as well as anything that is attaching the rebar to the burlap.
We've also asked, will this study be continued upon to monitor the further impact to the ecosystem, like in the future?
I That is a great question for both the tribe and the Lake Association. I hope they carry on. But yeah, there have been talks, but I'm not totally for sure on that.
Another question, were the macrophyte compositions in the reference plots fairly similar to the overall lake-wide aquatic macrophyte species composition?
Um, Lake-wide, I didn't use, so I looked at past surveys and it seemed pretty consistent with what I was seeing. Um, but so I want to say yes, but there was like, you know, my one site had maybe pretty much predominant Valsinaria or eelgrass, while the other site was a lot of native milfoil. So there were some differences there. But again, like comparatively to like other lake-wide surveys that I've seen from the past, whatever I was finding was consistent, like seemed to match up pretty well.
Thank you. And a couple more
Yeah.
questions. Do you think that the oxygen loss in the treatment areas may also have been due to decomposition of the pre-existing vegetation under the burlap, in addition to whether the weave was covered with sediment?
I do. Yes, that is another, yes, I do believe so. I think that if I was able to keep that DO logger underneath that newer barrier longer, that we would have seen that for sure. Because I could only keep him in for 2 weeks. Um, and it was also kind of scary to leave those things in there and find them again. But yeah, so if we had had him in there longer, I think we would have seen a decrease in oxygen once those plants really started to deteriorate.
And then was the native plant community that began to repopulate the treatment sites similar to what you saw in your nearby reference sites, or did certain native species recover more quickly under those post-treatment conditions?
Yeah, so they were similar to those native sites that we saw, but there were two that very much, were there first, and that would be the eelgrass and the pondweed. They seem to really come in there strong, like those were the first ones we saw come back.
Well, thank you, Anna. That was a lot of
Yeah.
rapid fire questions. Is there any other questions for the group? Otherwise, we can wrap up. Okay, it looks like that might be all of our questions, but I will be sending out an email with the link to the recording in the next couple days here. And Anna has kindly agreed to answer any additional questions people might have over email. And I just want to remind everyone that after the holidays, which I hope you all enjoy, that you join us for our next webinar on January 13th. That will be with the Northwest Michigan Invasive Species Network, and you can learn more about how you can partner with your SISMA to manage manage invasive species on your property. Okay. Thank you, everybody, and thank you so much, Anna.
Yeah, thank you guys.